Hello everyone. So the next topic which we will be going to study is about the next plant hormone, which is known as cytokinins. So in these particular slides we will be studying about the history of cytokinins, its structure, its function, how the biosynthesis and transport taken place in the plants, then about the psychological effects such as morphogenesis, apical dominance, then chloroplast maturation and senescence. So all these things we will be covering in these slides. So first we will get to know about what is the plant hormone. So this is a standard definition, that is naturally occurring organic substance that profoundly influence psychological processes at a low concentration. So in this figure basically all the plant hormones are being depicted and they are able to exert their action locally or at a distance. So next will be what are the types of plant hormones. They are auxin, gibberellin, cytokinins, ipsasic acid and ethylene. So our main focus here will be on the cytokinins. So what is a cytokinin? How it was developed? What was the first thing that the scientists came to know about? So the first cytokine was isolated from autoclave herring sperm in 1955. So this was an experiment which was being conducted by the scientist Miller and his associates. So they isolated from the first cytokinin was isolated from the autoclaved herring sperm. So this compound was named chitin because of its ability to promote cytokinesis. What is cytokinesis? It is basically a physical process of cell division which divides the cytoplasm of a parental cell into two daughter cells. So what the first naturally occurring cytokinin was isolated from corn by Miller and Lentham. Independently it was called zeatin. So in the next slide we will be showing the three structures of chitin, zeatin and isopentyl adenine. So these are the three structures which are being depicted here. So they are basically N6 substituted adenine derivatives that have diverse effect on important psychological functions in the plants. So they play a major function in the plant, and these are basically the three structures, the adenine structures that are being depicted here. In the next slide we will be discussing about the functions of cytokinin in the plants. So in plants basically the cytokinin will lead to cell division. There are root and shoot meristins. There is a cell differentiation because of cytokinins. There is a leaf senescence. The role of senescence will be discussed in detail in the next slides. There is inflorescence growth. Then there is a nutrient mobilization. Then there is a cotyledon expansion, apical dominance and seedling morphology. So some of these functions will be discussed in the slides that have a psychological effect on the plants. So in the next slide we will be discussing about the biosynthesis and the transport. Cytokine biosynthesis happens through a biochemical modification of adenine. So basically naturally occurring cytokinins are adenines which possesses either an isoprenoid or aromatic side chain at the N6 position. So in the structure slide the three structures of cytokinins are being shown. So the most common and active form are zeatin and isopentyl adenine. So cytokinins are generally more commonly they are found in higher concentration in the meristematic regions and the growing tissues. They are believed to be synthesized in the roots and they are translocated via the xylem to the shoots. So cytokine biosynthesis happens through the biochemical modification of adenine. So what it happens is it happens because through the biochemical modification of adenine. Its biosynthetic pathway starts with the formation of isopentyl AMP. So the reaction, the first pathway of this starts with the formation of isopentyl adenine by the removal of the phosphate group. So when the phosphate group is removed, it leads to the formation of isopentyl pyrophosphate and AMP. So isopentyl AMP is converted into isopentyl adenosine by the removal of the phosphate group. This was the removal of the phosphate group. Here the phosphate group was removed. Then after the removal of the phosphate group, isopentyl adenine is further converted into isopentyl adenine by hydrolytic removal of the ribose group. So the next step was this. Then finally isopentyl adenine is oxidized to zeton by the replacement of one hydrogen by a OH in the methyl group. So there are active forms. Cytokinins are represented are also present as ribosides, ribotides or glycosides. So these are three active forms in which the cytokinins are present. So this is how a biosynthesis and transport of cytokinin in plants takes place. The first basically the formation of isopentyl AMP is the first thing which leads to the formation. Then comes the isopentyl AMP gets converted into isopentyl adenosine. Then it gets converted into isopentyl adenine, and finally it gets converted into zeton. And these are the three active forms of the cytokinins. Then what are the psychological effects? That is cell division, morphogenesis, apical dominance, chloroplast and maturation and senescence. So we will be discussing in detail about all these processes. So first comes is the cell division. So cell division is involved in the formation, maintenance and growth of the shoot apical meristems. So what happens is cytokines act as a positive regulator of cell division in the shoot apical meristems. So what happens is cytokinins act as a positive regulator also and as a negative regulator also. So in case of positive regulator, cytokinins acts as a positive regulator in the cell division in the shoot apical meristem, and it acts as a negative regulator of cell division in the root apical meristems. So what happens is they are involved in the regulation of G1S and G2M transitions. So this is how a cytokinin play a role in the cell division. It acts as a positive regulator also, and it acts as a negative regulator also. So next slide is talking about the morphogenesis. Cytokinins and auxins stimulate morphogenesis, that is shoot and root initiation in the callus. So when auxin, for example another hormone, plant hormone which is auxin, when it is present alone, the ratio of auxin to cytokinin is high and the callus will initiate the root formation. And if a high cytokinin to auxin ratio is there, then it will promote the shoot formation. So roughly if we take the equal amounts of cytokinin and auxin, it will promote the proliferation of undifferentiated callus. So in this a morphogenesis plays a role. So a high cytokinin to auxin ratio promotes the shoot development, and equal amounts of auxin to cytokinin ratio promotes the proliferation of undifferentiated cells. And if the auxin to cytokinin ratio is high, then it will lead to the root formation of the callus. So this was all about the morphogenesis and how the cytokinin to auxin ratio plays a role in the shoot development, root development and proliferation of undifferentiated callus. So just we have to only see that where this is high cytokinin to auxin ratio, there will be a shoot development. When there is a ratio of auxin to cytokinin ratio is high, that will lead to the root development, and equal amount of auxin and cytokine promote the proliferation of undifferentiated callus. So both the cytokine and auxin, they induce crown gall formation in the dicot plants. What is crown gall? It basically originates from the plant tissue that has been infected by the bacterium, agrobacterium. So this was about the morphogenesis part. So the next we will be talking about the apical dominance. So there is a direct application of cytokinins to axillary bud which inhibits the apical dominance and it stimulates the growth of the lateral bud in many species, which leads to the overriding the inhibitory effect of the shoot apex. The cytokinin, what does the cytokinin does is that it inhibits the BRC1 genes. BRC1 is the genes which is being inhibited by the cytokinin and prevents the auxin-induced stylogactin biosynthesis. So this was about the apical dominance and how the role of cytokinins is played in the apical dominance. It basically inhibits the apical dominance and stimulates the growth of lateral buds in many species. So next we will be talking about the chloroplast maturation. Chloroplast maturation, it basically promotes the conversion of ethelioplast into chloroplast via the stimulation of chlorophyll synthesis. There are a dark ground seedlings which are said to be etiolated. The internodes of etiolated seedlings are more elongated and chloroplasts do not mature. So instead of maturing as chloroplasts, the protoplastids of dark brown seedling developed into ethelioplast. Which contains protochlorophyll instead of chlorophyll, and they do not synthesize most of the enzymes and structural proteins required for the formation of the chloroplast. So when seedling germinate in the light, chloroplasts mature directly from the protoplastids present in the embryo. So this is how a role of cytokinin is being played in the chloroplast maturation. Basically, cytokinins promote the conversion of ethelioplast into chloroplast via the stimulation of chlorophyll synthesis. So the next we will be studying another effect, psychological effect. Psychological effect is about the senescence. So what is senescence? Senescence is basically an energy dependent genetically programmed aging process which leads to organ plant death. Basically it is a programmed aging process which leads to organ and plant death. This is an essential process of plant's growth and development. It is a very essential process for the development of the plant and the growth of the plant. As plant tissue senescence, they produce enzyme necessary to recycle materials and reroute the subunits to areas for the use by active growth elsewhere. So cytokinins, what role does cytokinins play in this? There are many hormones which act as a positive regulator for senescence, while others act as a negative regulator in case of senescence. But in case of cytokinins, they cause delay in the leaf senescence. For example, if we do an activity, if we take a small spot on a leaf and it is treated with cytokine, we take a leaf and we take a small spot on the leaf and treat it with the cytokinin. That spot will remain green after surrounding the tissues on the same leaf begin to senescence. So the spot which was being treated with cytokinin will remain green, while the other part of the leaf will senescence, that it will lead to program aging, but the area which is treated with cytokine will be green for a long period of time. So the concentration of endogenous cytokines decreases in most senescing tissues, and exogenous application of cytokines cause delay in the senescence in the most tissues. So the mechanism by which cytokinins are able to delay the senescence is not very clear. There is not much study which has been done, but there is some evidence that cytokinins exert a role in mobilizing the nutrients. So one possible explanation for the senescence delaying effects of cytokinins is the enhancement of zinc activity in the plants. So tissue with the greatest cytokinin contents are the strongest metabolic sinks, and thus the majority of the nutrients are directed to them. For example, in some fruits, the onset of senescence is marked by the transient rise in the respiration. So there are other hormones also which is responsible for senescence. For example, there is abscisic acid, there is ethylene, which is responsible for senescence. But cytokines, they also play a main role in senescence by leading to delay in the leaf senescence. So this was all about the senescence. So these were the references from where I have taken the content and have made the slides. Thank you.